Greetings to you all. Greetings. This is Teacher Dr. Mgombo Ronald, MBCHB, BS Mark. Now, we are going to go ahead and look at the genetic code and the process of translation. Previously, we looked at, if I'm to just take you quickly, we looked at the central dogma previously in our previous video, where we said in the central dogma of molecular biology, we look at the movement of information from the DNA through transcription into RNA, from RNA to protein by a process known as translation. And we said that deviations from the central dogma, which we call reverse transcription, where we see in the presence of reverse transcriptase enzyme, we use RNA as a transcribe, as a template to form DNA strands in the retroviruses. We said those are exceptions from DNA from the central dogma. We saw the different types of RNA, and we said we have three different types of RNA. We have messenger RNA, which is made in the presence of an enzyme known as DNA polymerase II. Then we said ribosomal RNA, which is made in the presence of an enzyme known as DNA polymerase I. And then we said transfer RNA, which is the smallest among all of them, which is made in the presence of DNA polymerase III. And we said, we talked about the functions of each of those different RNA molecules, where ribosomal RNA acts as a catalyst when it comes to joining of the amino acids all together and then bringing the codons into proximity with anticodons. Now, we said we went ahead and looked at the differences between DNA and RNA, where we said DNA has ribose sugar, RNA has ribose sugar, DNA has deoxyribose sugar. We went ahead and looked at the bases, where RNA is usually made up of uracil, yet DNA is made up of thiamine. In that area, we said usually RNA is usually made up of a single strand, DNA is usually made up of a double strand. We went ahead and said that RNA is usually short, but DNA is usually very long. We went ahead and looked at the process of transcription. In transcription, we talked about the different components of a transcriptional unit, and we said it has a promoter region where the RNA polymerase binds. It can either be at the negative 35 upstream 5' end. It can be at negative 10 5 upstream, where we say negative 10 is the Tatat box. Then it can also be at positive 1 when it comes to the prokaryotes, to the eukaryotes. Now, I went ahead and said that it also has the transcriptional gene, that gene that is going to be read, that is going to be transcribed. And then it also has the terminator gene. We talked about the whole process. We said transcription is made up of initiation, elongation, and termination. Where we say termination can be row dependent or row independent termination. Where in row dependent termination, we said it is where the raw protein comes and kicks off the RNA polymerase. Then raw independent, the RNA polymerase goes off in the absence of the raw protein. Now, I went ahead and said that the heteronuclear RNA is not yet active, it is inactive, so it undergoes processing. And the processing is done in very many different ways. We usually say, we said it involves adding a 5' cap, adding a cap at the 5' end, where we said it is methylated guanosine triphosphate, which is usually added at the 5' end. We added a polyadenyl signal is added at the 3' end of the heteronuclear RNA. We said we splice out the introns, and sometimes we also do alternate splicing, where we, in case we want to form different enzymes, in case we want to form different proteins from the same gene, we just change the arrangement of the exons. Like for instance, when we are forming different types of antibodies, IgA, IgM, IgG, IgI, we just change the arrangement of the exons. We went ahead and looked at, so the capping, we talked about it already. We talked about the introns being regions within the DNA that can code for a protein and do not appear in the final. So they do not code for any protein and they are removed. So we talked about that already. We saw the process of splicing. We looked at it in details, where we remove the exon, the introns, which are the intruders. we saw how everything is done, and finally we are having our final messenger RNA ready to be taken to be used as a transcribe to form DNA, to form proteins during a process known as translation. So today we are going to go ahead and look at the process of translation. So that is what we are going to go ahead and look at, the process of translation. So when you look at the process of translation, as I told you earlier, in translation we are going to go ahead and have a messenger RNA being converted into a protein. This process of converting messenger RNA into a protein is usually accompanied by a number of things. One, we need the requirements for the process of translation to take place. We need to remember that one, we need enzymes. As we are going to see, we have enzymes like amino acid transfer RNA synthase, amino acid transfer RNA synthase enzyme, that is playing a very important role when it comes to activation of the transfer RNA. We have other enzymes, as we are going to see, translocase enzyme. So we are going to look at different enzymes. We also need we need transfer RNA molecule, we need amino acids, we need ribosomes, we need ATP, and we also need magnesium. This magnesium is very important, as we shall see, that this magnesium is very important for holding the small subunit of the RNA of the ribosomes with the large subunit. So they are held together by the magnesium. The ATP is very important when it comes to activation of the transfer RNA molecule. The transfer RNA molecule usually undergoes activation in the amino acid transfer RNA synthase complex. In our body, we usually have what we call the amino acid transfer RNA synthase complex. This complex is usually, they say they're about, I would say they're about 20 different complexes, all having binding sites which are usually specific for a given amino acid and for a given transfer RNA, whereby that is where the amino acid goes and then it is fused with the transfer RNA in the process known as activation. During activation, we usually see an amino acid being added on the onto the 3' end of the transfer RNA in the presence of ATP. So ATP is usually hydrolyzed. So when you look at the structure of the transfer RNA molecule, it is usually clover shaped. In 2 dimensions, you look at it as a clover shape, and when you look at it closely, it possesses an anticodon. You can see this area here, it is called an anticodon. So an anticodon is usually a triplet of nucleotides which are complementary to, it is usually complementary to the messenger RNA. It also it shows amino acid acceptor site, so it has an amino acid acceptor site at its 3' end. So the 3' end, which is usually having an unpaired CCA. The 3' end here has an unpaired CCA. So this area here, which is having unpaired CCA, those bases, they are like the binding site of the amino acid. So the amino acid comes and binds there. So for every amino acid, there is a specific transfer RNA for it. You cannot have one transfer RNA carrying more than one kind. Okay, as we are going to see in characteristics of genetic code, we shall see some exceptions whereby one transfer RNA having we cannot find one transfer RNA having a certain anticodon carrying more than one type of amino acid. Each amino acid is specific to a specific transfer RNA. We are going to see the factors. So there is no transfer RNA for a stop codon. There is no transfer RNA for a stop codon. We are going to go ahead and see that. So as we are going to see, so there are a number of things that we shall need. When you look at amino acids, they form the raw materials of protein synthesis, and we say there are about 20 of them. There are about 20 of them, as we shall see, and they are available in the cytoplasm. I talked about the importance of ATP. I told it is very important for activation of this transfer RNA to form the amino acyl transfer RNA, which is the activated transfer RNA. You can see it here. You can see that this transfer RNA, sorry about that. When you look at this transfer RNA, it has the D arm, which is very important for binding, for fusing, for holding this transfer RNA within the ribosomes. And it has also this other arm, which is very important for identification. This arm here that I'm touching here, it's very important for identification. Or the A arm. This arm, it is the one that is usually identified by the amino acyl transfer RNA synthase complex. And we have seen the anticodon arm, which has the anticodons, which are complementary to the codons of the messenger RNA. We have said that this area here, which is the binding site for the amino acid, it is usually having an paired CCA sequence. It is like the receptor site. So ribosomes, we shall look at the ribosomes serve as sites for protein synthesis, and each of the ribosomes is made up of two subunits, the small subunit and the large subunit. They usually occur separately in the cytoplasm, and it is only during protein synthesis that these two subunits get associated together in the presence of magnesium ions. So magnesium ions are very important. So the RNAs usually serve as immediate molecules between DNA and the proteins, as we saw earlier how they are formed. So now we can go ahead and look at the whole mechanism of DNA translation. So in prokaryotes, usually the ribosomes bind to specific translation initiation sites. These can be several different initiation sites of the messenger RNA. So we can have very many different initiation sites of the messenger RNA. A prokaryotic RNA can code for several different proteins. Translation begins at a code. This code here, known as AUG, it's a code that codes for methionine. In fact, now this methionine, for them, they have a special type of methionine known as N-formylmethionine. That is for prokaryotes. And then the same code can code for methionine in eukaryotic cells. So N-formylmethionine is for prokaryotes, and then methionine is for the eukaryotes. So that is the starter. So in the eukaryotes, ribosomes bind at the 5' then move downstream along the messenger RNA until they reach the first code known as AUG. So the code which codes for methionine, I've talked about that already. So translation starts when at that point. So I talked about the activation process already. Now the large subunit. So when now the ribosome is moving, remember the large subunit of the ribosome joins with the small subunit, and that requires magnesium ion in it. So the initiator codon, we have talked about it. So now what happens is the initiator charged transfer RNA occupies the P site. As we are going to see, when you look at this ribosome, you can see that now you can see that the small subunit has bound. The small subunit has bound here to the sequence of amino acids, the sequence of nucleotides, which is AUG, the first one here. So what happens? The large subunit also comes and binds, but it binds and this is held together by the magnesium ions. When that happens very well, now you should remember that this AUG is usually going to be correlating with the P unit, the P site in the ribosome. And then now we are going to bring in, after that has happened, now we are going to bring in the activated amino acyl transfer RNA, or what they call the charged transfer RNA. But this charged transfer RNA molecule is going to bind in the P site, and it is carrying an anticodon which is complementary to the AUG. The anticodon that is complementary to AUG, and along with it, it has carried amino acid methionine. I told you in prokaryotes, the amino acid now that it would be carried here would be N-formylmethionine. But here in eukaryotes, we have formylmethionine. We have methionine just. So that is the whole process of initiation. Having looked at initiation, I think we are now ready to go ahead and look at the elongation process. So during elongation, we have a number of items which we usually do. So usually the ribosomes have two sites for transfer RNAs, called the P site and the A site. The initial T site, which is attached to the amino acid in the so usually the transfer RNA is first put in the P site, and now a new transfer RNA with another amino acid comes in and attaches in the A site. It enters in the A site, it binds in the A site, and then the ribosome catalyzes the transfer of this amino acid from the P site into, now we are going to transfer this, the first one which was carrying methionine, we are going to transfer it into the E site. We have 3 sites, we have 3 different sites. So if I'm to take you back here, so you can see that we have the E site here, we have the P site here, we have the A site here. So now the new one that is coming in here, so during elongation, as you can see from this illustration, activated amino acid is usually added at 1 by 1 to the first amino acid, that is to say methionine. Amino acid is usually activated by using energy. We talked about that. Now, the addition of the amino acid occurs in 3-step cycle. The codon recognition, we first recognize the codon, and then the amino acid transfer RNA molecule enters the ribosome at the A site. The anticodon binds with the transfer RNA. Now, the amino acid on the first, now what happens now? We have said amino acid, now the second one is going to enter the A site. The first one, methionine, was at the P site. Now the second one is entering the A site here, as you can see it here. So amino acid of the first initiator is in the P site. I've talked about that. Now the second one enters the P site, the A site. Now the A site joins by peptide bond. So the amino acid in the A site is bound with the amino acid on the P site by peptide bonds. However, enzyme ribozyme act as an catalyst. Remember I talked about ribozymes. So ribozymes, this is where they act. Ribozyme is the catalyst for the process of forming the peptide bond between the amino acid in the P site and the amino acid on the A site. So at this time, the first transfer RNA in the P site is kicked out through the E site. So now the ribozyme is now moving in the 3' direction. Now it is moving towards the 3' direction. So when we kick out the amino, after the peptide bond has been formed between the amino acid in the A site and the amino acid in the P site, then this amino acid, what happens next is what we call the translocation. So the T amino acid at the A site carrying the dipeptide now at the A site moves into the P site. This process is called translocation, and it uses energy. So in translocation, both the subunits of ribosomes move along in relation to the transfer RNA and the messenger RNA. So what we usually see here is the transfer RNA carrying the dipeptide now gets into the P site of the ribosome, as you can see it clearly here. Now the one that is carrying the messenger, the one that is carrying the dipeptide is now in the P site. You can see it here, making the A site vacant. So since it is making the A site vacant, at the same time, the next charged transfer RNA molecule carrying the amino acid will now be received. During this process, the first uncharged transfer RNA is displaced from the E site. You can clearly see that here. Now when you look at, now this process is repeated as the amino acids are added to the polypeptide, and it takes just less than about 0.1 seconds for the formation of the peptide bond. Remember, the formation of the peptide bond is catalyzed by the ribozymes. They are the ones. So the third charged amino transfer RNA with the amino acid arrives at the A site. You can see it here. And then the anticodon and the codon bind by hydrogen bonds. And when they bind by hydrogen bonds, the polypeptide bond is formed in the influence of the ribozymes. The second transfer RNA is discharged from the P site to the E site and leaves the ribosomes. So the events like arrival of the transferring transferring amino acid complex, formation of the peptide bond, ribosomal translocation, and removal of the previous transfer RNA, which is now uncharged. Keep on repeating, it repeats itself. So as the ribosome moves along the messenger RNA, all the codons on the messenger RNA are exposed by one by one for translation. So that's what usually happens until, so that is elongation. That is what usually happens during elongation. Now let us go ahead and see what terminates the whole sequence. When do we terminate the whole sequence, and what causes it to get terminated? And that is now where we are at this time. Now, so we usually have three codons, which are known as the terminator codons. And as this ribosome is moving along from the 5' end of the messenger RNA to the 3' end of the messenger RNA, when it is moving, when it reaches and meets these three codes, these codons are known as the terminator codons. These codons are known as the terminator codons, or the release factors. They are very important. When they are met, they are the ones which will switch on the process of, which will switch off the process of translation. They are UAA, UAG, UGA. I usually like abbreviating them as you are away, you are gone, and then you go away. I repeat, you are away, that is UAA. You are gone, that is UAG. You go away, that is UGA. When that stop codon is exposed to the S site, it is not read or joined with an anticodon of the amine of any transfer RNA that is activated. So the release factor binds to the stop codon. When the release factor binds to the stop codon, then thereby terminating the process of translation, the polypeptide is now released into the cytoplasm. So now when the polypeptide is now released into the cytoplasm, you can clearly see that the release factor is binds to the stop codon. You can see this is the release factor. So it comes and binds to the stop codon. When it binds to the stop codon, we shall release the polypeptide into the cytoplasm. And the two subunits of the ribosome, you can see that they are separating. Remember, they were held together by magnesium ion. So now they are released into the site, they dissociate, and the last transfer RNA is set free to the cytoplasm. So now the messenger RNA also has some additional sequences that are not translated into proteins. And those untranslated units, by the way, they are found at the 3' end. Those units are called, the untranslated units are the ones which are usually abbreviated as UTR. The UTR, those UTR regions are the untranscribed regions. The UTR are usually present at both the 5' end, that is to say before the beginning of the start codon, which we called AUG, which codes for methionine in the eukaryotes, and N for methionine for the prokaryotes. Finally, the messenger and it's also in even after the transcribable unit, that is to say at the 3' end, we usually find them there. They are required for the efficient translation process. So they must be there. They're like telomeres in the DNA. They are very important for efficient translation to take place. So they are always there at the 5' end and the 3' end, the untranslated regions. So usually finally now we see the messenger RNA being released into the cytoplasm, and then it gets denatured by the nucleases immediately. Hence messenger RNA is short-lived. It does not live for so long. So that is the whole process of the translation. So allow me to go ahead and talk about the genetic code. So let us close up our discussion by looking at the genetic code. Now, The genetic code is the list of all the different codons and amino acids that they code for. In simple terms, that is what we call genetic code. The genetic code is a sequence. It's just a list of all the codons and amino acids that they encode. That is what we usually call the genetic code. And this genetic code, the main features of the genetic code were proposed in genetic experiments which were carried out by Crick and his colleagues. And they said translation starts from a fixed point, and that fixed point, we already talked about it. That is AUG, which codes for methionine. And they went ahead and said there is a single reading frame maintained throughout the process of translation. That is to say, we read from 5' to 3' direction. Those codons, when you look at them, the way they are read in the 3' to 5' direction. They are read in the 3s, and they are read in the 5' to 3' direction. That does not change. And they are saying that each codon consists of 3 nucleotides. And when you look at these codons, all of them, let's see them here. So you can see these codons, they are in 3s. They are in 3s, 3, 3, 3, 3. They are made up of 3. Now we are looking at the elements of genetic code. They are 3 nucleotides, 3, 3, 3, 3. That's why they call it triplet. And it has a distinct polarity. I've talked about that. They can be read, when you look at the properties of genetic code, 1 triplet, 2 distinct polarity. That is to say, when you are reading, it is in the 5' to 3' direction. And each code is triplet code. And when you look at the next property here that we are having here, we are having non-overlapping. When they say genetic code is non-overlapping, they mean that the genetic code only each base is part of only one codon. And an adjacent codon do not overlap. Each single base is part of only one codon, and adjacent codons do not overlap. If overlapping would happen, then we would be having about 6 conservative bases, only 2 amino acids. So this code, to remind you, it is non-overlapping. And then another thing that we can add on, we can say that genetic code is degenerate. What do we mean when we say genetic code is degenerate? Genetic code is usually degenerate because usually a single amino acid is encoded by single codon. However, some amino acids are encoded by more than one codon. Take an example of cysteine. Cysteine has 2 codons that code for it, while isoleucine has 3 of them. This is what they call degeneracy. And degeneracy is brought about by what we call the Wabble effect or Wabble hypothesis. In the Wabble hypothesis, we usually see the first 2 bases of the codon are usually the same, but the last one is the one that usually differs, or the one that varies. When you look at isoleucine, we can take an example of cysteine or isoleucine. So when you look at them, you realize that it is different. Or let's take an example of phenylalanine. Phenylalanine has UUU, UUC. All of them code for phenylalanine. Now, you can see that the difference is only in the third one, where we are having either U or C. That is what we call the Wabble effect. When you look at genetic code is also referred to as universal. What do we mean when we say universal? We say it is universal simply because it is used for both prokaryotes and eukaryotes. Wherever you will go, whether bacteria or a human being, AUG will always code for methionine. However, when you look at some variants exist mostly in the mitochondria, where we have very few genes. For instance, when you look at CUA codes for leucine. CUA codes for leucine. But at the same time, it codes for leucine, so it is universal code. But in yeast mitochondria, it codes for threonine. Similarly, AGA codes for arginine, and it is universal. But when you look at in human beings and in Drosophila mitochondria, it is a stop codon. So there are some exceptions that we can talk about. So we have been looking at genetic code. I told you genetic code is degenerate. You can see what the meaning of degenerate. You can see here you can see leucine here is coded for by 4 different amino acid, different codons. That is to say, CUU, CUC, CUA, and CUG. And the only difference exists in the third position. That is called Wabo effect. This is very important in preventing mutations. In case there is a single mutation happening, or a point mutation happens in the third position of the codon, then that will not change the protein that will be formed, because these other we shall still form the what? The amino acid. It will be placed in the rightful correct order. That is the importance of the Wabo effect. It can try to prevent the Wabo effect. We have also said that when genetic code is triplet, there are series. We have also said genetic code is non-overlapping. We have also said genetic code is universal. We have also said that genetic code is having codons known as the stop codons, which we have mentioned and said that is UAA, UAG, and UGA. You are away, you are gone, you go away. They terminate the process of translation. Genetic code is non-ambiguous as well. Why do we say genetic code is non-ambiguous? We usually say specific amino acids are coded for by a particular codon. Alternatively, 2 different amino acids will never be coded for by the same amino acid. To conclude, when you look at genetic code is a set. There are about 64 possible codons, but they only code for 20 amino acids. Remember, of those 64, we have 3 stop codons, and it is only 61 that code for an amino acid. Genetic code can either be 1, triplet, 2, has a distinct polarity, that is to say it is read in the 5' to 3' direction. Genetic code is commaless, genetic code is degenerate, genetic code universal, genetic code is non-overlapping, genetic code is non-ambiguous, and we have also stop codons. You are gone, you go away, you are away. Thank you so much for listening. If this is your first time on the channel, please consider to subscribe. Please, even if it is the second time, consider to like, consider to drop in your comments for improvement of the channel. Have a nice day. Till next time. We have been looking at genetic code and the process of translation. I remain teacher Dr. Ronald. We meet next time. Thank you so much.